Chris. Thanks so much for joining me on this episode of Concentrating on Chromatography. I appreciate it.
Oh, thanks for having me.
So we hear a lot about antimicrobial resistance. So how serious of a problem is this right now?
It is really a seriously growing problem globally, right? We have rising antibiotic resistance against a broad number of pathogens that were once easily treatable by clinically available antibiotics, and we are rapidly running out of tools in our arsenal to combat these resistant organisms, and we also have a shortage of new antibiotics coming onto the market.
And where are the current antibiotics falling short in just general terms?
So the ones that are clinically available, right, we only have a couple antibiotics, things such as vancomycin as kind of our last line of treatment for the resistant strains. We also have rising resistance in tuberculosis strains. And then in terms of drug development, there's not, from the big pharmaceutical companies, there's not a large push for new ones because the time frame of them being commercially profitable before resistance sets in is kind of a bottleneck towards us getting newer, more effective treatments. And the ones that are coming out on the market are more repurposing of existing drugs, kind of pairing them in combination therapy with something to help overcome resistance, or basically a new evolution of existing antibiotics that are targeting already known antimicrobial targets.
Now, just as a warning, I'm going to butcher this term, but
Okay.
for listeners like myself who are totally new to this, what is a peptidoglycan and why is it critical to bacterial survival?
Okay, so peptidoglycan, kind of think of it as the skeleton of the bacterial cell, right? So it is a glycoconjugate, right? So it is composed of a carbohydrate backbone, right, of alternating of two sugars, right, N-acetylglucosamine and N-acetylmuramic acid. So that forms this long glycan backbone, and then adjacent glycan strands in the cell wall are cross-linked via peptides that are linked to that muramic acid residue. So it basically builds this three-dimensional mesh around the cell that basically serves as the skeleton, right, confers shape to the bacteria, and it confers shape and is also responsible for cell growth and division, right? So that peptidoglycan has to grow continuously without defect that could result in lysis of the cell.
And if I understood it correctly, most antibiotics target cell wall synthesis. So how does this differ from that?
Right, so the currently available antibiotics and historically targeting, so it's a great target for one because it is unique to bacteria, right? So it's something we can target bacteria with hopefully little cross-reactivity to the host, right, the human host, right? It's still an excellent target because really for the most part, most of the antibiotics to date have focused on biosynthetic steps in the cytoplasm or in the cross-linking steps, so creating that three-dimensional mesh by cross-linking those peptides in peptidoglycan strands to create that mesh. And there has not been not a ton of focus on looking at other enzymes and components of that very complex pathway that one can target.
And in this paper, you focus on enzymes that degrade the carbohydrate backbone, let's say.
Yeah.
What made that class of enzymes the right place to study?
Well, always a little bit of luck, right? Factors into everything, right? So one of the features when we started thinking about this is, right, the carbohydrate backbone of peptidoglycan is fairly invariant across all bacteria, gram-negative and gram-positive, right? And it's relatively intolerant to modifications, right? So conversely, the stem peptide, where probably the most famous antibiotics, the penicillins, act on, right, by preventing cross-linking, there's a lot of variability in that stem peptide, and a lot of the biosynthesis things, other amino acids get substituted in that may help to confer resistance. right? Whereas that glycan backbone, well, it hasn't been targeted in the past, right? Very little tolerance to any modifications present, so that was kind of our impetus for trying to go after that class of enzymes.
And you developed small molecule probes and inhibitors. What was your strategy for designing them and what made them effective?
We use something that we refer to as aglycone profiling, right? So we were interested, and this is done with really productive collaboration with a longstanding colleague and friend down at Brown University, right? We were interested in one particular class of enzymes that target the carbohydrate backbone of peptidoglycan, referred to as N-acetylglucosaminases, right? That basically cleave that sugar backbone to create small soluble fragments. And so to try and target that class enzymes, what we did was we took a non-carbohydrate component, or our aglycone, right, and we linked it to a sugar, right? So in our case, N-acetylglucosaminases recognize and cleave the N-acetylglucosamine glycosidic linkage in peptidoglycan, right? So we used the sugar to hopefully target our class of enzymes that we were interested in, right? And then we employed a top-down strategy, right? So in one, we were like, oh, we're really interested in maybe targeting these enzymes, but we're also just interested in finding potential any inhibitors, and we'll deal with the target ID later on, right? So we did a top-down where we started with whole cell assays, which also kind of helps our selection process for other properties that are amenable to potentially a small molecule drug, right? Being able to get into the cell, being effective, all that type of stuff. Did that screen, and then we kind of whittled down basically like a lot of phenomenology, phenotypic kind of screen, right, to get us down and kind of hone in on what we might be inhibiting, right, in the cell. And then actually it was one of my former students that was kind of working in the lab one day is like, oh, we should really test this particular glucosaminidase and see if it's the inhibitor, and like it ended up being correct, right? So we're working our way down towards it, and he's like, oh, I think it might be this one, and we moved on from that. So it's been kind of an iterative strategy, right? So we started with a class of molecules referred to as glycosyl triazoles, where we have a non-sugar component linked to a sugar, and then we've been evolving these molecules ever since by kind of cutting off the sugar and kind of modifying and evolving the non-sugar aglycone component to try and find more potent inhibitors.
And you mentioned that top-down approach. Is that how you validated that these probes were actually engaging the intended enzyme in the biological system, or how did that validation process work?
So that validation process kind of, we initially started with basically like a whole cell screen for just looking for things that halted bacterial growth, right? So we screened a range of organisms, gram-negative, gram-positive, and then from there, once we identified hits that were inhibiting growth, we then kind of started doing a series of genetic screens, looking at morphology changes under the electron microscope, looking to see the changes, try and kind of pinpoint the things that our compound might be interrupting, right? And kind of hone our way down in terms of narrowing down to class. So we would then start with some whole cell basic enzyme assays, just looking to see what activities we were inhibiting, and kind of then whittling down to our final protein target.
And in your results, you showed changes in the actual fragment profiles. What exactly are you looking at, and what were the key signals that told you the enzyme was being inhibited?
Yeah, so we've been using LCMS approach for analysis. So that's kind of investigating kind of the changes that are occurring upon treatment with our compound. Right? We knew we were targeting the cell wall. We were looking to see how the cell is adapting upon inhibiting our given target protein. And right, so that's taking insoluble peptidoglycan. So to be able to analyze it, we have to digest it to make it soluble, right? And what we're looking for there is basically changes in cross-linking patterns. So are we seeing more monomers of disaccharides of peptidoglycan versus dimerized cross-linked or trimers and tetramers, right? So basically, in terms of from the LC standpoint, looking at changes in retention time. So the more cross-linked the fragments, digested fragments are, longer retention times observed, right? Right, so that's a quick eyeball. You can look at the chromatogram and go, okay, we're much longer retention times. We have more cross-linking, but we're also looking at also changes to the backbone. So the few modifications that are tolerated in bacteria are things like O-acetylation of the muramic acid and N-deacetylation of the N-acetylglucosamine. So we can also observe changes in those profiles as well. So kind of we're inhibiting something that targets the carbohydrate backbone. We're looking to see how the cell was responding to those effects and kind of pinpoint that mode of action.
In this podcast, we focus on separation science broadly, but obviously how LCMS fit into your workflow was one of the main things I was interested in learning about. And it seems like in terms of what you were actually measuring, that it was very much that focus on retention time, as you noted.
Yeah, retention time, and then also because peptidoglycan not very, in terms of classic HPLC, not very sensitive in terms of not a good chromophore, right, for absorption. So we use LCMS to also kind of pinpoint what exactly those changes represent, right? So looking at intact mass analysis of the fragments coming off, right, to determine, okay, well, we're dealing with a tetramer here based on mass, and then if we need to, we can go to higher resolution mass spectrometers and do a little tandem mass spectrometry and kind of fragment to kind of get a little bit more detail there as well.
And what did the sample prep look like prior to the LCMS?
Well, so dealing with trying to analyze peptidoglycan can be difficult because it is this insoluble polymer. So we actually exploit that insolubility in terms of its isolation, right? So we can basically solubilize everything into detergents except the cell wall, and we can do differential centrifugation to pull that out. Then we have to do a number of purification steps, so enzyme digest to remove contaminating nucleic acids and proteins. We also need to remove covalently linked teichoic acids in the cell wall. That's like the other major polymer in gram-positive bacteria is these teichoic acids, which complicate LCMS analysis, right, if we don't remove them, right? So we remove those either with HF treatment, or now more often we do a short HCl treatment to remove those. And then to actually be able to run on an LCMS, we have to digest, right? So we do an enzyme digest of that insoluble peptidoglycan, and we use an enzyme that actually gives us a little bit of information about the structure itself, right? So that the enzyme we use is mutanolysin. That enzyme will only cleave the peptidoglycan if the next muramic acid in the chain is not cross-linked, right? It has to have an intact pentapeptide side chain. So we get a little bit of information from the cleavage pattern that we get from the enzyme that we use, right? And then to reduce complexity of the LCMS, we also have to reduce all the reducing ends so we don't get a bunch of diastereomeric peaks, right, present on the chromatogram. So we have a little bit of borohydride reduction, and then spin columns to remove our digesting protein and any contaminants before loading to the LCMS.
Yeah, sample prep. Although I was certainly well aware, just based on what Organimation does, sample prep is obviously one of the most complicated parts of the process, depending on what you're focused on. And it sounds like there's a lot of potential pitfalls just with sample prep alone.
Yeah, no, there's a lot of spots where you're not careful and not following between samples exactly the same procedure, right? Artifacts can kind of crop up in the process, right? So you want to be careful that you're always kind of isolating, prepping in the same way, kind of using the same tools so we can have a good direct comparison.
Now, whether focused on sample prep or possibly the entire analytical workflow, let's say, what makes this so challenging in terms of, are you dealing with similar structures, low abundant species, or just complex mixtures in general? It sounds like there's complexity all over the place.
There's complexity. And from an LCMS standpoint, some of these like tetramers and stuff are low abundance, don't necessarily fly particularly well, right? And then from a classic chromatography standpoint, the chromophore is not very great, right? We're basically looking at amide absorption, right? At around 218 nanometers, so that's not particularly sensitive. And then the complexity all comes in with the bacteria, right? Even in the same bacteria, there can be some changes into that. The glycan backbone is constant, but there can be some changes in the stem peptide, right? In terms of what's there. There's a substitution of an amino acid, so you can get low level substitution of another amino acid that will complicate your analysis downstream.
I wanted to rephrase this next question slightly. What are the downsides to using LCMS for this workflow? Is there anything you learned the hard way in terms of common pitfalls of this LCMS application?
The common pitfalls is making sure that it starts before we even get to the LCMS, right? And that we're doing everything consistently between treatments and samples that we want to analyze, so that we're always always run a control that we bring along the process every single time to make sure that the control and the treat. So we're always comparing one to one every time, just to deal with whatever like biological systems. So they vary, right, from growth phase to growth phase, and so making sure that we're always doing things consistently up front so that we can be very reproducible in our analysis downstream on the LCMS.
And how would you say this work can translate into potential new antibiotics? And are these enzymes realistic drug targets?
Potentially, I mean, there's been a little bit more in the last few years interest in targeting these classes of enzymes, and there's been actually a natural product a few years ago that was identified that broadly inhibits a broad range of these enzymes that are involved in the degradation of the bacterial cell wall, right? So I think kind of a lot of what we're doing is kind of a little bit of like using our compounds not just as like potential antibiotic target, but as a chemical probe to kind of study the function of these enzymes, a little bit kind of providing orthogonal approach to the classic genetics and molecular biology approaches that have been used to study these enzymes, using our probes to kind of tease out a little bit more of the role that these enzymes play in growth and division. And then some of the stuff that we're working on now, right, not only we're maybe validating our existing compounds as maybe a viable target or a viable hit that can be pursued, but also through our kind of chemical biology approach, we're in the process of identifying potentially new targets for antibiotics that could also be exploited downstream for new antibiotic development.
And one of my favorite questions that I like to ask researchers, if you had unlimited resources, let's say, what experiment would you run next, or how would your workflow change to try and better answer some of the questions?
That got me thinking, and one of the things if we had the funds and everything, kind of some using our probes to and in tandem with some proximity labeling mass spec techniques for proteomics and starting to map out our interactome type of thing. I think that would be a really interesting series of experiments, right? And kind of might lead to some really interesting questions.
I think the concept of antimicrobial resistance is something that everyone, including non chemists, have heard of or come across. What is something about that topic in general that most people would misunderstand?
I think that in terms of the importance of antibiotic resistance, I think is that how our arsenal of available clinically useful antibiotics is dwindling quite fast, and the push to develop new ones or to repurpose or reestablish existing ones by targeting the resistance mechanisms is not keeping up, right? So in that battle with the bacterial pathogen, the pathogens kind of got the lead at the moment.
And Chris, we certainly covered a lot today as a general introduction to your work, but Was there anything that we missed, or was there anything else that you would like people to know about your work that we didn't talk about?
Yeah, we're kind of a chemical biology. We kind of do everything from synthesis in the lab right through to biochemistry to, right, I got students in the lab that do very heavy microbiology projects, and molecular biology is the kind of all of the, we kind of use all those tools and together to try and get to that answer. Kind of our overarching philosophy in lab is trying to use chemistry to kind of solve biological problems.
No, that's very cool and something I certainly hope to learn more about in general. Yeah, Chris, thanks so much for the time today. This is a super important topic, and obviously, I was thrilled to be able to connect it to your use of LCMS. So it's been super interesting to learn from you. Thank you.
Thanks so much for having me. It was a really great opportunity.